Welcome to the Explainer. In this one, we're taking a deep dive into Corynebacterium macginleyi, an organism that's been quietly moving from the sidelines of microbiology right into the clinical spotlight.
So let's get right into it. We're going to look at how this one bacterium, which was only first described back in 1995, has basically gone from being this overlooked bug to a really serious concern, all thanks to better diagnostic tools and its pretty alarming drug resistance.
Just so you know, everything we're about to cover comes from a Gemini deep search. If you want to see all the source materials for yourself and really dig into the data, the full notebook document is available. All right, here's our game plan. We're going to walk through this just like a clinical case. We'll start with why it's a threat, then cover how to spot it in the lab, what it actually does to people, how we treat it, and finally, how to stop it from spreading.
Okay, first up, why is Corynebacterium macginleyi suddenly on everyone's radar? Let's break down how it made the jump from what we thought was just a harmless colonizer to a pathogen we really have to respect.
Okay, and this slide right here, this is where things get really serious. Just look at that resistance profile. We're seeing widespread, high-level resistance to both fluoroquinolones and macrolides. And this isn't random. We know it's because of mutations in the GIR gene and the presence of the Ermax gene. But, and this is a big but, look at vancomycin. It's still susceptible. You're going to want to remember that because it becomes the key piece of the puzzle when we talk about treatment.
So with a resistance profile like that, you absolutely have to know what you're dealing with. There's no room for error. This brings us to identification. And thankfully, our lab methods have come a long, long way.
Okay, back at the bench, the #1 giveaway for this bug is its lipophilism. It just loves lipids. You'll see these tiny little pinpoint colonies on a standard blood agar plate after a couple of days. You might even miss them. But add a little bit of a lipid source, something like tween 80, and boom, they just take off, growing into these big, robust colonies. Sometimes you'll even get this really distinctive rose-pink pigment. It's a fantastic little trick for a presumptive ID.
Now, if you're using something like an API Core 90 strip, there's one reaction that's a dead giveaway: the negative pyrazinamidase. That one little negative result is what really helps you separate Corynebacterium macginleyi from a lot of its look-alike relatives.
And this right here is so important because it highlights a huge potential pitfall in the lab. If you're just relying on 16S rRNA sequencing, you're going to get stuck. The sequence is way too similar to its cousin, Corynebacterium acalens. You just can't tell them apart. To get a definitive ID, you have to sequence the RPOB gene. It has a lot more variation, and that's what makes it the gold standard here.
Of course, the big game changer in modern labs is MALDI-TOF. And honestly, it's a huge reason we're even talking about this organism so much now. It's something we call diagnostic unmasking. We're finding it more because our tools are better. But, and this is the crucial part, MALDI is only as good as its library. If Corynebacterium macginleyi isn't in that spectral database, the machine might misidentify it or just call it a no ID.
Okay, so we figured out how to identify it, but what does it actually do to a patient? Well, it turns out this organism's reach extends way beyond just a simple eye infection. We're talking about a spectrum of disease that can be pretty severe. Its primary turf, so to speak, is the eye. It's actually the most common corium bacterium we find on the conjunctiva. But we've learned it's not just sitting there harmlessly. This is a true pathogen. We now know it produces this powerful exotoxin that directly attacks corneal cells and kicks off a major inflammatory response. It's not an opportunist just piling on, it's the one starting the fight. And this is where Corynebacterium macginleyi becomes a really serious nosocomial threat. In those vulnerable hospitalized patients, especially the immunocompromised, this thing is nasty. Why? Because it's an expert at forming biofilms on things like catheters, breathing tubes, and prosthetic joints. And that biofilm-forming ability is what turns it from a localized problem into a cause of life-threatening systemic infections.
All right, so you've got your positive ID, you know the patient is sick. Now what? How do you actually treat this? And this is where it gets tricky, mostly because of that drug resistance we saw earlier, and, frankly, a lack of official standardized guidelines.
Yeah, this is the million-dollar question for both the lab and the clinicians on the floor, isn't it? We don't have species-specific breakpoints from EUCAS for Corynebacterium macginleyi. That means we have to borrow breakpoints from other bacteria, like staph or strep, to interpret our results. It works, but it adds this layer of uncertainty and really highlights why the microbiologist and the physician need to be talking to each other. So, let's look at how some folks are handling this. The Dutch guidelines give us a really logical framework. For a serious infection, you start with what you know works, IV vancomycin. You hit it hard and fast. Then, once you get those susceptibility results back from the lab, you can de-escalate if possible, maybe switch to a high-dose penicillin. But if it's resistant, you pivot to linezolid. It's a smart, data-driven approach.
And that same logic really holds up on an international level. The consensus is pretty clear. For any serious infection where you suspect this bug, vancomycin is the backbone of your therapy. Your main alternatives are linezolid and daptomycin. But remember that crucial detail about daptomycin. It gets inactivated by lung surfactant, so it's a total non-starter for pneumonia. The main takeaway here is don't gamble. With the high rates of resistance, you really can't afford to start with anything else empirically.
When it comes to kids, the principles are pretty much the same, but everything requires more precision. All your dosing is going to be weight-based, and you'll definitely want to get an infectious diseases specialist on board. Vancomycin and linezolid are still your go-to drugs. And just like with adults, given their resistance, fluoroquinolones are off the table. Okay, we've covered how to identify it and how to treat it. That brings us to the final, critical piece of the puzzle: prevention. How do we keep this opportunistic pathogen from causing infections in the first place?
The key thing to understand here is that there's no fancy, high-tech solution. It's all about mastering the fundamentals of infection prevention and control. That means obsessive hand hygiene first and foremost. It means using contact precautions for patients with multi-drug resistant strains. And, maybe most importantly, it means being incredibly disciplined about how we manage medical devices. Aseptic insertion and getting them out as soon as they're no longer needed, that's huge.
You know, we'd really love to hear from you. If you've encountered this organism in your own practice, share your experience in the comments below. Your insights are so valuable. And of course, if you found this helpful, please like and subscribe so you don't miss our next explainer.
Which brings us to one final thought to leave you with. When you consider its growing multi-drug resistance, its talent for forming biofilms, and the fact that we're only now getting really good at finding it, you have to ask, is Corynebacterium macginleyi the next major hospital-acquired threat that's been hiding in plain sight this whole time?